Hi folks, today we're going to look at solving Punnet squares. A Punnet square is a diagram that we use to determine or predict the outcome of a genetic crossing between two organisms. There are 4 easy steps to follow. Let's look at them quickly. Step number 1 is to determine the genotype and the phenotypes of the parents. Now we know that genotypes is expressing the genes as a letter, capital letter or lowercase letter, and the phenotypes is to physically say what the parents look like. If we look at the example here, for instance, two capital D's means that the parent is homozygous because of the 2 identical alleles being present, and dominant because of the 2 capital D's for that specific trait. If we look at this one, 2 lowercase or small d's means that the parent is also homozygous but recessive because the parent has 2 identical recessive genes for that specific trait. The parent can also be heterozygous, which means that both alternative alleles for both traits are present in the parent. Right, we are going to step number 2, set up the Punnet square with 1 parent on each side. Now if we scroll down, we can see here is an example of such a Punnet square, where we can see on the top we show the male genotype, in other words the letters allocated to the male, and on the left we have the female genotype, or the letters that we allocate to the female. Right, so we write those letters that represent the specific alleles on the side and on the top of the Punnet square. Right, let's scroll back up. Number 3, once we've done number 2, we are going to complete the Punnet square by filling it out. And then once we've done that, we will look at the different genotypes that are in the Punnet square, and we will determine the possible ratio or percentage of the offspring. Good, let's look at this little example here. In pea plants, round seeds are dominant to wrinkled seeds. So that means round seeds are dominant and wrinkled seeds are recessive. In order to determine the letter that we're going to use, we look at the dominant trait, round, and we say, right, we are going to give a capital R to round seeds and a small r to wrinkled seeds. What possible genotypes and phenotypes can we get out of these 2 traits? Well, the genotypes can be homozygous dominant, that means 2 capital R's, and that will mean that we will have definitely round seeds. We can have homozygous recessive, which means that we will definitely have wrinkled seeds, and we can obviously have the heterozygous capital R and small r, which means that we will have round seeds because of the dominant trait being present. Right, so genotypes gives us the alleles that are present in the reproductive cells of the parents, and also the phenotypes tells us what that feature or trait looks like in the parent. Right, we have here an example. In radishes, a bent root is a dominant trait, although some roots in the radishes may be straight. So some radishes may have straight roots and some radishes may have dominant bent roots. Right, so the bent root is dominant, so we're going to choose a capital B there, and the straight roots are recessive. We're going to have a small b there. If the straight-rooted plant is crossed with a heterozygous plant, how many of the offspring will have straight roots? Now that's the percentage. how many of the offspring will have straight roots. Right. So we have a straight rooted plant, right? So it's straight roots. And since straight roots is recessive, we know that both alleles present in the parent must be recessive. So that's why we have got the lowercase b's there. And then there is a bent root roots, and that's a heterozygous plant, and we are going to say, right, capital B, small b. Good. If we go to our Punnet square, now we don't know what the male, which one belongs to the male and which phenotype or which characteristic belongs to the female, so we're just going to write a capital B, small b, and small b. Right, just how do we get to that? If we look at the cells from which the reproductive cells are formed, upon meiosis, that cell divides and it produces two reproductive cells. And the alleles split and then appear in each of those reproductive cells. If we look at the other one, obviously we will find the following. That both reproductive cells will have the recessive gene. Good. Number 3 says complete the middle of the square. Right, so if we go and pair the male and the female alleles, we are going to find from the male we have the dominant allele, and from the female we have the recessive allele. Right. And if we do that for the next one, dominant allele from the male and the recessive allele from the female. If we go to the next column, we can see there is a recessive allele and another recessive allele, and if they pair up, we have two lowercase b's, and exactly the same will be for the other one also, the other square. So we analyze these numbers and we determine what is the phenotype and the genotype of the offspring. If we go back to our initial question, we need to find out how many of the offspring will have straight roots, right? So now we can say, right, we've got two squares, heterozygous, so that means 2 of the 4, that's 50% of the offspring will have the dominant bent roots, and we have here 2 squares with a recessive trait. That means 50% will be the recessive straight roots. And our answer here would be, of course, 50% of the offspring will show the recessive straight roots due to the genotype of the offspring being lowercase b, lowercase b. Right, that was pretty easy. That's the end of this video. Have fun practicing. Thank you.